A bunch of wonderful keynote speakers today. We're going to start out the first keynote talk by Dr. Jennifer Listgarten. She's a senior researcher from Microsoft Research. Thanks. So we're going to start out the first keynote talk with Dr. Jennifer Listgarten from Microsoft Research across the river. Jennifer's current interest in computational biology is actually a very interesting trajectory to that point. She started out with an undergraduate degree in physics and then did a master's in computer vision, and then finally a PhD degree in machine learning at the University of Toronto. And her current focus is on machine learning and applied statistics with applications to medicine and biology, specifically genetics, immunoinformatics, proteomics, and DNA sequencing, and so forth. And I can say more about it, but I would highly recommend her recent interview that she gave to the Talking Machines. If those of you who have not heard of it, Talking Machines, just Google it. It's an interesting website where they interview researchers in data science and machine learning, and she shares her personal journey into this domain and also how she brings biology and machine learning together. So on that note, I'd like to invite, please join me in welcoming Jennifer to give the talk.
Thank you for a lovely introduction. So I'm going to give you a whirlwind tour of two topics that I've worked on recently, just to give you a flavor of how machine learning and biology mix. But I just wanted to say, in case you are wondering who am I, what is Microsoft Research? So we're actually just across the river from you, so I guess we can actually look at you and you can look at us. And it's a very multidisciplinary lab, and among some of the areas are computational biology and machine learning, which is exactly what I do. So, I'm going to sort of do three main parts, and one is going to be some motivation and some basic terminology. So when you do biology, there's a lot of terms to learn, and I'm going to try and give you the minimal ones that you need to understand this talk. And then I'm going to tell you about genome-wide association studies, and which is about finding sort of the genetic underpinnings of disease from large cohorts of genetic data. And finally, I'm going to close with some more recent work on actually gene editing and how you can use machine learning to improve gene editing. So first of all, why do we study these things? So I'm sure you've all heard of the Obama initiative for precision medicine, for example, which is adapting treatments to a person's genetic makeup. So these are the reasons we want to collect this kind of large-scale genetic data and analyze it. There's also things like risk identification. So if you can figure out ahead of time what people are sort of predisposed to, you might hope to catch it sooner and somehow improve their lives. And then also, sort of medicine has always been very not very data-driven, I would say more observational. And so a lot of diseases are not sort of very cleanly defined. And now that you have access to large-scale molecular data, you can actually hope to more cleanly define a lot of these diseases, and that's called disease subclassification. And then finally, just looking at this kind of data, often you get insights into just basic mechanisms of disease and how the body works, which is useful for a variety of things. So now let me walk you through a little bit of terminology. So what's shown here is called the central dogma of molecular biology, and if you're an information theorist, you'll be pleased to know the information goes in one direction here, and basically it goes from the genetics up here. So this is your DNA, the two chromosomes, the helices intertwined on one chromosome. And you'll see these little green things are meant to be mutations at a single point. And so I've highlighted this and made it very big because we call these SNPs, and I'll probably forget that I'm not talking to biologists and keep talking about SNPs. So when you hear me say SNPs, just think genetic marker where there's a mutation in the genome. And so, we all have them, and it explains our various susceptibility to diseases and to curly hair and to being tall or short. Anyway, so from there you get transcription, which gives you this sort of RNA. And I think you may have heard some talks on gene expression, and that's measuring the body sort of at this level. And from there, it gets translated into proteins, which are sort of the workhorses in the body that really do a lot of things, and ultimately cause some, what we call is like a phenotype, or if you're a machine learning person, you might say the target variable. And so that would, maybe be you have a cardiac disease or you don't, or you have curly hair or you don't, or what is your blood pressure. These are all referred to as traits or phenotypes. And so what I'm going to tell you quite a bit about are genome-wide association studies, in which you're looking for associations between these genetic variants and some target or phenotype of interest. And so now this might sound very simple, like I've just told you about all this biology, and now we're focusing in on this very narrow problem. And so it is, biology is very complicated, and so people tend to break these problems apart, and there's people working on different parts of it, and there are people working on putting it all together. And I'm going to tell you a sort of little vignette about this particular kind of analysis, although I've worked on many other kinds of data as well. But I think there's an interesting story in terms of machine learning and statistics, well, for all of them, but especially this. But so the grand goal is to merge these things all together, and I'm going to tell you about a kind of narrow slice and a story about that. So, right, genome-wide association studies. Let me just make sure we all know what I mean. So the input here is a set of people, say, with and without a disease. And then imagine like 500,000 people. In the old days, you were really happy if you got 100 people. And so, it's ramping up very quickly. And so what you do is you measure a large set of genetic markers, say roughly 1 million right now is a pretty standard thing to do, for all those people in your cohort. And then what you do is you can sort of step along each one of those markers on the genome and say, is this associated with my disease of interest? Of course, what you really want is causality, and I'm going to gloss over that here because it's a more complicated story. But in genetics, you know that the gene affects the phenotype. So if there's an association, you know at least the direction. It's not going the other way around. And even though these are not causal arguments, you can extract a lot of causality from this kind of data and analysis. So once you conduct an analysis like this, what you get is a Manhattan plot, or that's one way to visualize it at least. So what's shown are the 22 chromosomes here, and someone has, for example, for blood pressure, gone through the genetics of people and figured out which individual markers on the genome are associated with blood pressure. And the higher the dot, the stronger the association. And so, right, this is a very simple analysis. And if you're a statistician and you're not a geneticist, you're probably laughing, wondering why they do this. But again, there are reasons to do this, and you can do more sophisticated analyses. But for the purpose of my story, it's going to be fine to talk about this just one test at a time. And so people have done this for quite a few years now, and there are catalogs online of all the things that are known. This is quite an old one. And there's a very rich body of literature and catalogs and databases where you can find a lot of information that's been gleaned from these things. So what are some of the statistical challenges here? So as you might have guessed, there's a very, very many hypotheses we're going to test, right? So I said basically 1 million markers or of these SNPs on the genome, and for each one we're going to compute a p-value and ask if it's significant. And if you start to do that many, many times, as you do with 1 million SNPs, then you have some very large probability of many of them looking significant. And so that's one challenge, and I'm not going to address that one. A second one that I am going to address, it's what's called data heterogeneity. Or that's one thing you can call it. Another way to think about it is that you sort of have hidden factors in the data that you don't know about that are going to wreak havoc with your analysis. And so an example of that is if you have family relatedness, so people, grandparents and parents and siblings, that little cluster of people are more related to each other than to the rest of the cohort. And similarly, you can think of sort of racial diversity in a similar but larger scale way. And it turns out that with standard statistical analyses, this just really screws things up. And that's been the focus of a lot of my work in the past few years. And so, but sort of the flip side of these challenges is that when you have all of this data, you can actually, in this particular instance, you can kind of see problems in the data if you look at it in the right way. And I'm going to give you an example of that. And you also have enough data to infer these hidden factors that are wreaking havoc with your analysis. And so, so there's some wins to be had here. So right. I've mentioned these sort of hidden factors that are going to mess things up, and that's what I'm going to tell you about is kind of how can we model them and make them go away, and how can we figure out by looking at plots if we have made them go away. So the fundamental assumption in most models, if you're not a statistician, is called sort of identically and independently distributed. What that means in this problem is basically that this cohort of people you have are kind of all roughly the same. So they're not in these little clusters due to family structure or due to racial variation. In fact, they were just sort of very homogeneous, and you never get a data set that's homogeneous, and so what happens is you end up with these spurious correlations, and I'll tell you a little bit about how that happens in a second, but basically all kinds of markers start to look like they're causal for the disease when they actually are just sort of tagging things like race and relatedness. So this is going to take a minute at least to walk you through this plot, but I think it's quite fascinating because it's one instance of how the large scale of the data lets you diagnose problems if you have a few assumptions that you can make. So imagine that I'm walking through the genome and I'm getting a p-value for every one of like 1 million markers. If you, under the assumption that there is actually no genetic cause of the trait, so none of these hypotheses are actually valid, none of the genetics influence the trait, then what you expect to get is a very particular theoretical distribution of these p-values. In fact, it's uniform between 0 and 1. And so you know that. And so that's a really nice thing to know because in genetics, it's frequently the case that you assume that very, very few markers are associated with the disease. In fact, so few that you can kind of pretend it's this assumption that none of them are. And so what you can do is you can plot your observed p-values against your expected p-values in a sort of quantile plot. And if you see a large deviation, then you know that you've actually screwed up the analysis. And in this particular area I'm talking about, it can be a very egregious violation. So this is real data, I haven't made this up. And what you see is that if there were no signal, it should follow the diagonal line between the error bars, but it actually is way out here. And these are, I should say, these are minus log p-values. So this is highly, highly significant out here. And like I said, this is real data using linear regression, which is a sort of standard off-the-shelf way that you would think to analyze this. And so, but it's nice because now we know that something's wrong, right? There's some caveats, like you need to be careful. If you have a large enough sample size, like getting to like close to 1 million, and you have for particular traits, it could kind of be valid, not quite this extreme, but on the whole, this is a red flag. And so on the other hand, this is real data, and this is a model I'm going to tell you about, basically brings it back to where you expect it to be. In this particular data set, you don't really expect signal, and it's here. If there were signal, you would expect a few points out here, but the bulk of it to lie between those lines. So how are we going to fix this problem? So as you might imagine, the basis of the problem was this sort of uneven similarity between people in the data set. And so if you could somehow model the pairwise similarity between every 2 people in the data set, then you might hope to fix it. And so that's precisely the starting point for any method that actually fixes this problem. So these are what I'll refer to as genetic similarity matrices. And what it is you take all of the genetics of a person and you basically imagine this room is the cohort. And what's shown here is, like, say, I don't know how many people are in this room, let's say 100. There's 100 people on this axis and the same 100 people there. And every entry is the pairwise distance between those people based on their genetics. So like you have a vector of all your million SNPs. I have a vector of all my million SNPs. You take the inner product, and that gives you some similarity. And it turns out that that's very, very useful. And I'll show you how that's useful. So first, if you do just a naive analysis, again, remember we're stepping through one marker at a time, and we're going to get a p-value. And so the naive analysis, again, is just linear regression, which assumes people are technically IID or sort of all equally dissimilar. And when you do that, that's the graph I already showed you. And so this is, people always, the next slide is the favorite slide whenever I give this talk, and it's not my work, but it's pretty cool. So the next thing people started doing is they said, take this matrix and use principal components analysis on this matrix. And that's going to capture sort of major axes of variation in this high dimensional space. So let me try and rephrase that another way for those of you who are not super statistically oriented. Imagine that you have this very high dimensional space that all the SNPs live in. So every person in this room lives in like a 1 million dimensional space, which is dictated by the 1 million SNPs you've had measured. It turns out that there's certain patterns in this space, which means that it's not really 1 million dimensional, that you could actually represent it very effectively with, say, just 10 dimensions if you knew which dimensions they were. And that's kind of what PCA does. In fact, it figures out those 10 dimensions, figures out where each person lies in each of those 10 dimensions, and then it adds it as a covariate. And so why does this work? So this is the really fascinating plot. So if you take this matrix, this is real data. This was published in Nature some number of years ago and independently by other people on other data sets. If you take this genetic similarity matrix and you do PCA on it and you plot just the first 2 components, and then you annotate it with the country of origin of those people, which is known in this case, then you kind of recapitulate the map of Europe. And so that's kind of astonishing. And that explains why when you do this procedure, that if one of the problems with your analysis is, for example, racial diversity, this correlates very well with geographic diversity, and therefore it stands to reason that this would do a good job. However, this doesn't do a good job for if you have sort of very fine grained structure, which is this sort of pedigree structure. And that's where these mixed models come in. And I'm just going to focus on the very last line here. And so what is this last line? If you, again, I can walk you through it even if you're not super statistically oriented. If I took out this term here, this would be nothing more than linear regression, written in a particular way, a multivariate way, where you have all the people's traits. Example, blood pressure, they're all in a big vector here. And then without this term, this is linear regression. And what the mixed model does is it basically says, this term over here is what assumes people are sort of equally similar or dissimilar, or technically IID. And what we're going to do is we're going to add another term here that specifies the precise way in which they violate that assumption. In a perfect world, if we could compute that, that's what we would put here. And we're going to take our best guess of what that is using this genetic similarity, and we're going to include it there. And it turns out that this does better than this PCA thing, because PCA remember, is reducing to like only 10 dimensions, and I'm not doing anything here to truncate the space. I'm keeping the full richness of this matrix. And so on this real data set, again, this is an old data set that's a little bit weird because it doesn't have very many genetic markers, but it's very good to play with these kinds of models because it's a super challenging problem. It has 1,300 people who are in families, so this sort of pedigree structure, and then it also has 3 different races here. Right. So I have spent quite a few years doing work on these models for this problem. And I don't want to take you all down like a really niche hole of what those are, and I'll just give you a few flavor of what that's focused on, what problems, and a little bit of intuition on how you would actually tackle those and how we did tackle those. So there's 2 main things, and the first way I got into this space was that I knew about these models, and these models are notoriously very, very expensive to compute. And not just in some theoretical way, like in a practical way. You have a data set, you can't actually run the analysis. So formally, they scale cubically in the number of people in the cohort, and they scale quadratically in the memory requirements. And so things that, software that were out there that implemented this would actually just blow up. In fact, on the memory, but if they weren't blowing up on the memory, they would never have finished. And so we started doing some work on reducing those things. And then once we started doing that work, we had some more insights about these models, as happens when you do research, and realized we could also make them not only faster, but also actually better at doing the thing we wanted them to do. And so I'm not like, I give whole talks on this, and so I'm just going to give you 2 slides of some of like a rough outline of what some of those things might be. So to speed it up, it turns out that there are these very expensive computations, which are related to the actual probabilistic form of this model, which is a large multivariate Gaussian, and that's what incurs these very expensive cubic operations. And they have to do with the size of this matrix here. So the more people you have, it scales really, really badly. It scales like cubically in this number. And it turns out with some algebraic tricks, you can move things around and you can actually get it it's an approximate model now, but you can get it to be only linear. And this is actually a really dramatic improvement. It meant that you could suddenly analyze data sets you couldn't analyze before. And in the course of doing that, we noticed some other things about the model that had to do essentially with, earlier I said what you want is this to precisely capture the way in which this assumption of homogeneity was violated. But you need an oracle to tell you precisely the way in which it's violated. And unless you have an oracle, all you can do is take your best guess of how you should compute this. And so we had some insights on how you could compute it better, which differed from what people had been doing in the community, and which also solved one open problem that people had been a bit puzzled about. So I'm not going to say too much more about that, because I also want to tell you a little bit of a story of fixing genetics. So what I've told you so far is finding out where there's a problem in the genetics by looking at large cohorts that have a particular trait. And so imagine now you know where these problems are, like what's the next step? So it could be a number of things, and one thing would be to just edit it away sort of in a therapeutic manner. And that's not quite where we're at yet, but we're rapidly getting there. So, right. Right. So gene editing, it's actually been a goal in molecular biology for quite a while. But you may have, and you'll see on my next slides that it's become really, really big news recently due to a new technology called CRISPR. And so, as I've just said, sort of if you knew which mutations were disease-causing and you had this really good ability to just go in and sort of cut and paste the genome, then you could fix actually a lot of problems, right? And so it's no surprise that people are tackling this. And so now I'm going to tell you about this new system called CRISPR, which is all over the place. And so, in fact, it started off, sort of in obscure journals, and then slowly making its way to like Nature and MIT Tech Review and The Economist, and the New York Times, CNN, and I don't know if you guys saw this cover on Wired last year, and this is quite funny. So this cover is about CRISPR gene editing, and it says there's no hunger, no pollution, no disease, and the end of life as we know it. So that's quite a statement. So that's why I decided to work on this. No, I actually decided before this cover came out, but. And then, so there was actually quite a backlash on Twitter about this, and so this cover has been quite mocked by now, and provided quite a few good jokes. So yeah. But so more seriously though, so it's not quite ready for this prime time gene editing, but there have been some very promising results in sort of model organisms related to fixing one of the receptors from which HIV you get HIV through, and a defect related to cystic fibrosis, and also corrections for muscular dystrophy. But these are not, this is not in a human solved completely. It's on the road there. And so right, it's not quite ready. And why isn't it quite ready? So there are two main problems. One is you need better on-target efficiency, and what that means is that if I want to go to this particular location in the genome and I want to edit it, then to the extent that I manage to get to that position and do the right thing, that's my on-target efficiency. And the second thing is off-target effects, which is that if I'm trying to go to this position in the genome, I better not also be doing weird stuff over there. And both of these things are currently problems, and people are working hard to to get solutions to these. And you could do that in two ways. So one is with improved laboratory protocols, which is not my expertise. And so my expertise is machine learning, and that's one of the ways you can do that. And so we've done some work on the on-target, and we're currently working on the off-target. And this actually just came out, I think, at the start, on Monday, this came out in Nature Biotechnology, actually, the on-target work. But I think it's a really fascinating system, and so as much as I love machine learning, I actually want to tell you a bit about the history of CRISPR, of where it came from, and it's also useful to understand the machine learning. And it's also a real testament to funding basic research, because for many years, there are actually these poorly funded bacterial researchers in dark basements with crappy labs, spending time wondering what these repeated elements were in the genomes of bacteria, and probably their colleagues were laughing at them. Well, it turns out that this is the core component of this gene editing tool. And so the way what it was, eventually they found out, and actually it was a yogurt company, Danone, that found out, of all things, because this happened to be protecting some of their yogurt strains against viral attacks and not others. And so they actually had a breakthrough paper in this field. And so what it is it's a natural immune system for bacteria. And the way it works is that if a bacteria is invaded by a virus, if it survives that attack, then it actually kind of clips out part of the bacteria, sort of takes a photo and stores it as what I'll call a viral memory. And then if it survives, now it has this memory of this attack, and if the similar virus, or the same virus rather, comes along again, then it can actually recognize it, and it deploys this, which is sort of a protein-based scissors, and actually just cuts it and disables it. And so that's the naturally occurring system. And so what people have done, how do they use this for gene editing? So I can't tell you all the details, but just to give you a sense, is basically instead of allowing these viral memories to arise due to being infected with a virus, they kind of say, you know what, I can just make up a synthetic viral memory of where I want those scissors to be deployed, right? I'm not going to wait to be invaded from a virus. I'm going to put this in a human genome. I'm going to decide I want to go over here, and I'm going to sort of synthetically put in this thing. And this thing, which I'm kind of calling a synthetic viral memory, it's actually called a RNA guide, because it's going to guide these scissors to the right location in the genome. And so that's sort of how it works. And now, how does machine learning come into this? I'm glossing over some things here in the interest of time, but roughly speaking, there are two problems in gene editing. Sometimes what you really want to do is just to turn a gene off And other times you want to go and change like an A to a T. Right now I'm just going to talk about turning a gene off, but a lot of what I have to say applies to the other problem as well. And so it just so happens that, say, a gene is like this long on the genome, whatever, in whatever units, and you need to deploy the system. So you could cut it here, you could cut it here, you could cut it here, and all these places, right? It turns out that this CRISPR-Cas9 system, it works more effectively in some places than in other places. And so if I'm some lab somewhere in the world and I want to disable this gene, I need to know which of these hundreds of places is actually going to give me my best bet to actually knock down the function of this gene. And people actually did not know how to do this, and so they couldn't really effectively just arbitrarily knock down genes. And so working with the Broad, we basically custom made like a smallish data set, in fact, of about 15 genes. And so for those of you who don't know biology, there's about 20,000 genes. So it's a very, very small number of genes where we systematically looked at every one of these positions and how well you could actually cut it and disable it. And then we built predictive models with that. And so we generated all possible of these guides for a small set of genes. We measured how well they perform, which is actually quite a lab endeavor. It takes months and months, a lot of money, and a whole bunch of people. Then we, on the machine learning end, we featureize these guides, and I'll explain what I mean very briefly in a second. And then we basically build, evaluated, and selected a good predictive model. In fact, this is the first time in my life I've written a paper where I didn't do any new machine learning. We actually just used canned machine learning here and did it smartly with like our training and our insights to get a state-of-the-art model. And now we can predict the efficiency of, for any arbitrary gene that's not in this training data set, of which part of the genome you should select. And before this paper even went to press, we already had about 20 people asking us for the prediction server and the code. And I've never had that happen with my work before. I've had it after it's published, but so there's a lot of people really, really want these tools, is my point. Right, and so let's see, I guess I'm almost out of time, so I'll just quickly say like little things that we did that were different. Like a few people had worked in this space, but not very many, and they made some very bizarre decisions that we examined and kind of fixed. So people were treating this as a classification problem. They were arbitrarily saying that 80% of things would fail and 20% would succeed. And so you had a nice 0-1, but it was sort of a fake sort of 0-1. And so we backed off of this assumption, making it into a regression model where we would predict real valued numbers, like on a continuum rather than just 0 or 1. And we investigated more, much richer models of machine learning. And featureization, as you might imagine. So what's featureization? Featureization is basically taking the DNA, like this little guide or this synthetic viral memory, and turning it into a bunch of numbers that you can feed to the machine learning algorithm. And this is sort of an art in and of itself, unless you use deep neural networks, in which you have a different art of sorts. And so it turns out that we found some features that were more important or were additionally important to what people had been using. And some things that people hadn't been using are like the thermodynamic features of the DNA. So what happens is that when you go into this double-stranded DNA, the system needs to pull it apart. And so depending on the properties of the thermodynamics there, that will be easier or harder to do. And so that's just sort of one example of things that can affect whether or not it will work and that you can capture with the machine learning model. So in the end, we now have this, it's sort of the methodology is published and there's a prediction server out there that people can use. And we're continuing to work on this off-target problem, which is actually more interesting in terms of the statistics and machine learning for reasons I don't have time to go into. But and finally some acknowledgments. So all the GWAS work was for quite a while with Christoph and David. And the CRISPR editing was with Nicolo, who's here in New England as well, and John, who's at the Broad Institute. So, and that's it, thanks.
So we have time for questions for Jennifer.
Do you want?
I'll start. There's always a little bit of latency in having the audience start asking questions, so I'll ask a first one. Thank you very much. This is really impressive. Tells me how much I need to learn about where the world is. So I'm actually, in your last slide, you said, well, this is now on Azure, which makes it accessible to anybody. And earlier in the talk, you talked about how if one is naive about using tools, you can make big mistakes when you talked about how you assume IID and things are not, et cetera. So the question here is, and it's sort of at the heart of data science, is as tools become more accessible, it's important to also know when they don't work and what makes them work, et cetera. Can you comment on this? For example, an undergraduate can start using this without knowing much about why it works or when it works?
I would say if there's an undergraduate who's managed to get CRISPR up and running and needs to use this, I'd probably trust that undergraduate to have enough sense to.
Why would you put it on Azure otherwise? You don't want it to make it available.
No, so what, I mean, there's a lot of, like, basically this CRISPR stuff, there's some people who are pushing the forefront of this research on making CRISPR better, and that's like John Dench and Feng Zheng and Jennifer Doudna at Berkeley. Like, these guys are pushing that to take it further and further. But then there's a huge community of CRISPR users who are basically have their own labs, and they're interrogating systems, and they're trying to fix diseases, and they're just using it. And so what those people just need, like, which is why they're already bugging us, because they sort of heard about it and they see that it works better, is they just want to say, I'm in my lab, and today I want to knock down this gene. And so where should I deploy my CRISPR? And without this, I'm going to take a guess of one of 300 places. Maybe I'll do 5 of them. I don't have like enough money or time to do more than that, and hopefully those will work. And now instead, they can basically rank those 300. So in that sense, there's nothing to lose, even if it's perfect. And you can use the model for things it wasn't intended for. So other organisms, other kinds of so the scissors. It turns out there's different variations of scissors and things like this. And we're working to actually generalize the model beyond that. But I think even if it's abused in that way, chances are they're going to be better for them than in doing nothing. So, but I agree with your comment. Like, I think where your comment becomes more important is where you have labs who want to build a model like this. And so if we release the code that says, here's how we built the model, then I think you're very right. And so that's a different end user. And we are releasing the source code, so people probably will start to do that. And I guess it's sort of buyer beware.
Thank you. I had a question actually about your mixed linear modeling for GWAS and so forth, and I was wondering, so you're using it primarily actually to control for confounding for hidden structure and so forth, and if that's correct.
That's correct in this work. I have some other work I didn't talk about in which we are actually interested in testing genetic markers and a whole bunch of them together, and we put those in a variance component and test its statistical significance.
Okay. No, I was just curious if you actually going beyond what you did, if your approach can be used actually to look for substructure that is phenotype-driven substructure, basically, not so much as using that component to eliminate the substructure, but actually to extract information about that substructure, for example, to identify subpopulations that might be relevant with respect to the phenotype that you care about. And I'm wondering if that approach can be used to that end.
So I have some other work I didn't talk about in which we use this to model something called cell type heterogeneity. And so there the problem is when you start, like what I talked about here, there aren't really cells. Like if you spit or you take blood or whatever, you get all the same genetics, but there's a whole bunch of other biology where the biology depends like very, very much on the cell type. And when you take some like cancer tumor and then you sort of like measure a whole bunch of stuff, it depends very highly on what cell types are in there, and this is a huge problem. And so you can, and we have some work on this where you can use these same models. And so what it actually ends up doing is kind of identifying the cell type composition of each sample. So I think that's kind of what you're saying. But the caveat here is that it's not very good at telling you exactly what they are. It's only good at eliminating the influence of it. So I think if what you're most interested in is actually what are these hidden structures, I think there may be better ways to do that than the linear mixed model. Because it becomes sort of a post hoc looking at the mixed model to figure it out rather than baked in directly.
I just ask that please introduce yourself before asking the question.
Ted Pudlik, Physics. Could you comment on your choice of Python as the programming language?
Yes, I can. I was once in love with MATLAB. I used it for a very long time, and then we broke up, and I tried a few things, and then I started working with people who were very gung-ho about Python, and I'm actually not a big hacker myself, so I'd rather go with the flow and make use of all my colleagues' code, and if they're using Python, then I'm going to start using Python. But I think it's great because it has a whole bunch of stuff baked in. Like it actually has far more than MATLAB, and it's more of a proper programming language. But I'm really not, like to me that's just a tool. I'm not sort of like, people will argue very vehemently of Python versus R and when whatever else, but I don't know, it's just been convenient.
I'm moved, but still Valentin Voroshilov, physics department. Well, we all know why so much effort put into study genomics, physiology. We talk about physical health of the nation, right? What about intellectual health? We all know that it's really big problem have an army of good STEM teachers. Maybe a cheaper solution to teach Watson how to teach math and and physics.
So, sorry, what's the question? I
said
honestly, that wasn't a joke. I actually didn't quite get it.
Well, I'm actually serious. Maybe Watson,
Yeah.
who can win.
And what about Watson? So, could you just repeat the question, or was it a comment?
No, it's a question. Maybe it's cheaper.
Yeah.
Would be to teach Watson,
Oh,
and then
I see.
of course other computers how to teach simple algebra,
did you read
geometry.
The Financial Times had a very nice piece on Watson last week, which basically said it's like overhyped, like marketing. So that would be my answer about. Watson, like Watson's kind of like the Google brain. Like there's a whole bunch of people in a company, and they market it under. Microsoft does it too under Cortana Analytics. So.
So maybe Cortana or Siri could help. I don't know.
I think
no?
it's.
Don't you think?
There are a lot of people working on very seriously on using computational tools for education, pitching it at the right level, testing them interactively to see where they have gaps in knowledge. So I think there's definitely a role for computation in education. I'm not an expert in it. I'm not sure I would want to be taught that way, but maybe I'm old school. But I don't think like in any time soon there's going to be like a Watson instructing a class kind of thing. That's the grand goal of artificial intelligence, is to have such a thing, and I think we're very, very far away from that. So.
Thank you.
I always get these things wrong. I'm Graham Foisey. I'm in auditory research and biomedical engineering. One of the challenges in my field, especially for using principal component analysis, is that we're dealing with highly nonlinear systems. And I know that that's probably true even operating under a central dogma assumption in molecular biology, especially with kind of post-transcriptional methylation action and other kind of epigenetic effects. And I was wondering, how well that's accounted for by some of the corrections that you worked on.
So it turns out actually that in these kinds of problems in genetics, that actually linear assumptions are actually really, really reasonable. And in fact, there are many people who want, trying to show that they're not, and it's very, very hard to do that. So I don't think that for the exact problem I spoke about, that nonlinear approaches are super helpful. And that's not to say there aren't nonlinear things happening, but they're really dominated by the linear stuff, so it's hard to find them. But when you start talking about mixing whole systems together, not this sort of isolated slice of a problem, then yes, that's definitely the case. But there are lots of, PCA is just like, that's the sort of, first year undergrad approach to dimension reduction. And so there's a whole host of things in statistics and especially in machine learning that do highly nonlinear. So I don't know, Dana Pear is a computational biologist at Columbia who does a lot of work on flow cytometry data, and they have adapted some of the very highly nonlinear embedding or manifold embedding stuff like t-SNE to the cases, to biology, and have shown quite clearly that the linear methods don't work. So I think there's a lot, there's a large toolbox of things out there you can use if you're in that space and you want to do dimensionality reduction. But I don't think in the problems that I spoke about that they would actually help very much.
My name's Kyle from biomedical engineering. I'd be interested, as much as you can comment, just on kind of the general approach for you with research at Microsoft. I see some of your works in collaboration with the Broad, and how much of kind of what you do with the computational biology at Microsoft is kind of intended to be deployed by Microsoft versus what is kind of like, as much as you can.
I can say, I'm not going to hide anything. They love it when we collaborate, especially with like great institutions like the Broad. They love it when we publish. It would have to be something very central to our current business model before they wouldn't want to do that. And And that happens in research, but it's not the everyday affair. And so they're super happy about this work with the Broad. And in general, like my goal is to work with outside academics and publish things. And from time to time that leads me to something that the company might care about, and I do some side little thing internally, but that's really, really rare, and it's the minority of the work. I probably shouldn't. I'm on camera, right? Shoot. Do I get editing rights to this? So there's a balance, but we're definitely encouraged to collaborate broadly and to push the state of the art in computer science, and also look out for the company and help the company move forward.
And I guess my question is, does Microsoft have like a push in the biology space, or is it on the computational side? I'm not familiar with anything Microsoft pushing on the biology.
Yeah, you wouldn't see them pushing anything, but let's just say there's a lot of people thinking about ways in which they might want to do that. Yeah.
Hi, I'm Shane from physics. Could you comment a bit more of that map of Europe? I don't know if you could provide any intuition for why that happened.
Yeah, yes. So there are actually theoretical geneticists who've published papers on why that happens, and I actually don't know the results, but you can give a very intuitive explanation, which is that the way people mate, probably more so in Europe than in North America, is based on a very local geographic proximity. And so genetics follows sort of mating, and if mating follows sort of a spatial pattern, then the genetics will follow a spatial pattern. And that pattern is a very low-dimensional structure, and so even though the genetics are very high-dimensional, it gets represented very well in this very low-dimensional space.
Are the axes, though, also like with north, south, and east, west?
I do not. Did it say that? Really? Wow, I'm impressed. Let's see, I don't know. Actually,
But I don't know if they were the real.
I assume they had to like do some flipping around. I don't know that it's like, I don't think it goes north from south, but I guess if mating happened more along latitude and longitudinal axes, then it would perhaps emerge. But this just says PC1, PC2. So, they might have had to flip it, orient it to get it to map, but I don't think it knows those things. It's not my work, I'm not sure, but I would be very surprised if it did.
Other questions? Well, if there are no further questions, please join me in giving a round of applause. And so we're going to have a short coffee break. There's going to be coffee to my left there. And please assemble back here in about 10 minutes. Thank you.